All right. Well, thank you for allowing me to talk, and I'd like to acknowledge the traditional owners of the lands on which we meet. All right, so I'm going to change tack completely now. So we've heard a lot about RNA and genes, and I'm going to be focusing now on chemistry. Specifically, I look at metabolomics, which is a broad kind of analysis of chemistry within cells. So I'm actually a microbiologist, and my passion is really about how microbes talk to each other through the use of chemicals. So, the way that they interact could be a positive interaction. They could help each other to grow, or they could sort of have this antagonistic effect. And so not only am I interested in how microbes talk to each other, but also how microbes talk to their hosts. So in this case, plant hosts. And so what we're interested in is kind of understanding so microbes might use chemicals kind of as a tool for invasion, and then the plant host might use chemicals as a mechanism for defense. So really what we're trying to do is understand, well, what's happening in terms of chemistry inside cells. So yeah, they promote sort of beneficial interactions or can be involved in pathogenesis. So I'm interested in a number of questions in terms of chemistry. Firstly, I wanted to be able to understand what the response of the plant host was during early stages of myrtle rust infection at the chemical level. My second question was really to understand if there was any chemistry that was associated with resistance. So is there a set of molecules that maybe the plant is producing that might inhibit rust infection or germination, and what these molecules are? And then lastly, I wanted to understand if we could focus down on these important molecules and see if they could be used as biomarkers for resistance. So could we select these molecules, look for them in plants, and then see or predict resistance of those plants? So the tool that we use is a process called untargeted metabolomics. So it's kind of like what some of the other speakers were talking about earlier in terms of transcriptomics, looking at all of the genes that are switched on inside the plant or inside the rust. But we're actually looking at that from a chemical perspective. So rather than just saying, okay, I want to know what this molecule is doing or what this molecule is doing, we try to take like a broad approach or a broad analysis. Unfortunately, the way that it works is we can't analyse all chemistry. We can't analyse all molecules because we're kind of restricted by the technology that we use and the tools that we use. So in this case, we wanted to look at water-soluble molecules, so probably molecules that are hanging around inside the cells. So it turns out that this kind of technology is already being used in a number of different areas. So in particular to look for plants or breeding, to look for biomarkers that are associated with resistant to plant disease or abiotic stresses. And it's also even used to analyse human disease as well. So to start with, we wanted to look at what happened during early stages of infection And so this was really, I did this with the help of Jeff and Louise from Queensland. And so we took Melaleuca quinquenervia plants and we analysed them before infection, 24 hours, 48 hours and 5 days post-infection. So the way that this kind of heat map works is that each one of these rows across is an example of one molecule that's been identified in the process, and the columns are individual samples. So if we look at the grouping at the top, we can see that there was a very strong chemical response to early infection, 24 hours after infection, by a number of molecules. And then that kind of early sort of chemical onslaught by the plant kind of died off again. Oops, sorry, pressing the wrong buttons. That kind of died back down by about 5 days. So it didn't matter what the phenotype of the plant was. So we compared susceptible, hypersensitive, and resistant plants. And among these molecules that we looked at, there was no kind of difference between the phenotype. So, you know, some of these molecules could be important if we wanted to understand what was happening during the first 48 hours of infection. And also if we wanted to use or be able to develop a method to detect early infection. So then we looked specifically at the differences in the plant cells before infection, so at that day 0 point, because we wanted to know whether there was some kind of chemical difference between the plants even before that infection had occurred. So it turned out that, again, we were looking at kind of specific molecules, and each row is a particular molecule, and some of those molecules were, I guess, different in the resistant compared to the susceptible and compared to the hypersensitive. So that tells us that if we could identify some of these key metabolites that are associated with resistance, that we could understand how a resistance occurs, but we could also then see if or we could develop a tool for detecting resistance before infection has occurred. And some of those molecules that we identified as being important were flavonoid compounds, but I should say at this point that a lot of these compounds would be unknown. So, I mean, we detected a lot of molecules here, but most of them we don't actually know what they are or what they do. In terms of prediction, the predicting chemistry is not as good as predicting genes and RNA sequences. So we have a lot of work to do in that space. So then we wanted to ask the question, okay, so if we can detect resistance before infection in Melaleuca quinquenervia, can we do the same thing in other species? So we looked at a few different species from different genera, and we compared the metabolites before infection to see if we could identify some of those same molecules that we identified in the Melaleuca. Unfortunately, because these plants are all so different, there was no, I guess, similarity between these molecules that we identified, but we did see the same kind of pattern, that there were molecules, so these are volcano plots, and the reds dots indicate molecules that are more abundant in the resistant plants, and the blue indicates molecules that are more abundant in the susceptible plants. So we did again see a pattern and a difference between susceptible and resistant plants. And so, unfortunately, as I said, those molecules are species specific. So we couldn't develop like a tool to identify resistant plants that would be, you know, one size fits all. But if there was a key species that would be interested in breeding efforts, then we could develop a very targeted tool for identifying resistant plants. I did notice that in some cases, there seemed to be a lot of molecules that were more abundant in the susceptible plant. So in my mind, when I started out on this, I kind of thought, well, what I want to really find is those molecules that are important in resistance. So what is that defense that's going on in the plant that's stopping that early infection by the rust? But I did identify that there were a lot of molecules in the susceptible as well, which was kind of, I found to be, you know, unusual, but maybe that makes me want to think my hypothesis that maybe there's something in the susceptible plants that are helping the rust to grow. I don't know, maybe you might have some other ideas, but that was one of my thoughts. So again, a lot of unknowns were identified, but of those ones that I did identify, they were belonging to the terpenoids, flavonoids, fatty acids, and carbohydrates. And again, there was no sort of pattern of these molecules in the resistant or susceptible. Those molecules seem to be present in both, or those classes of molecules, I should say. So we really need to find out more about what these molecules are doing. So in the last step of this analysis, I wanted to find out more about whether we could use metabolomics as a protocol for field sample testing. So we took some leaves from plants that were grown outdoors, and we stored them at room temperature for up to 24 hours, just to see whether those metabolites would be stable. And then, you know, they could be easily sent from the field to a testing facility and then used for testing for resistance. And we found that some metabolites were actually stable and so could be good candidates for testing for resistance. But we would have to, as I said before, look at specific species and develop specific protocols. So overall, we identified that infection does trigger a metabolic response in the plants, and some of those molecules are phenotype specific. We found that metabolomics could be used as a screening tool for resistance, and that it would need a species-specific approach. And that molecules were more abundant in susceptible, which might be important in inducing infection. So where to from here? So we really need to develop a robust metabolic protocol for phenotyping plants and characterising those unknown compounds. So because the technique we've been doing is like an untargeted, we have a lot of background noise from all the molecules inside the cells. So now we can develop a method to find the ones that we are interested in and look for those specifically. So we can pick those out and use those in a more targeted protocol. We plan to analyse other metabolomics methodologies to look at a broader range of chemistry. So I said we were focusing on more water-soluble molecules, but in the future we could look at volatiles, or we could look at water-insoluble molecules, so things like lipids. So those are examples of future analyses as well. And we're also looking at how fungi that are present on the leaf might be important in helping the leaf to become resistant or inhibit germination in the plant. So thank you for listening. And thank you to everybody who helped. I always forget the acknowledgement slide.
Wonderful. Okay, any questions for Michelle? Yes. Oh.
Is that working? Yep. Michelle, how localised is the change in chemical profile? Is it limited to the leaf or the particular axis bearing the leaf, or does it ramify across the whole plant?
You mean the
post-infection profile change?
We only tested the leaf, so we could look at that. That would be interesting to look at. But yeah, we're limited by funds and time. But definitely a good idea for future work.
Any questions?
Perry, you have a question?
Yeah. I just wondered if maybe the field test you did to see how durable the metabolites were, would you think about using those stable markers to reduce the noise of all the other ones that you could then...
Yeah, definitely. So the way that we, so the samples are run on a mass spec, and you know, the mass spec is kind of set up to analyse all metabolites, but you can actually use the mass spec to focus on those markers. So yeah, it would be the next step to just say, okay, blur out the rest of the noise and just focus on those, and then, you know, hopefully we would see the same pattern.
So you mentioned that the molecules of interest were species specific, but did you find that some of those molecules that were in the resistant phenotypes were then found in another species but in the susceptible phenotype, or were they like really separate?
No, they was definitely separate.
It's really interesting.
Yeah. So there was a lot of molecules that were consistent across all of my samples, but those that were specific to the phenotype were not consistent across